Hello, Assalamu alaikum. I'm Dr. Bushra, and the topic we are going to discuss today is neuron and neuroglial cells. The learning objectives for this lecture are that at the end of this lecture, you'll be able to name the various cells of the nervous system. You'll be able to describe the structure and function of neurons, discuss the axonal transport and its types, enumerate the various functional zones in the neuron. You'll be able to classify the neurons and enlist the various glial cells and mention their types. To start with, there are two types of cells in the nervous system. One is the neuron, also called the nerve cells, and the other ones are glial cells. They are also called the glue cells or the supporting cells. The neurons are the structural and functional units of the nervous system. They are excitable and they conduct the impulses that make possible all the nervous system functions. Neuron consists of three parts. Number one, the cell body and the processes. The shorter ones, the dendrite, and the longer one, the axon. First, we'll discuss about the cell body of the neuron. It is also called soma or perikaryon. It is irregular in shape. The mass of cytoplasm is also called neuroplasm. It is covered by a cell membrane, and the various contents of the cytoplasm are the nucleus and various organelles like the mitochondria, Golgi apparatus, nasal bodies, and the neurofibrils. The nasal bodies and the neurofibrils, they are found only in the nerve cells and not in any other cell. The nucleus in the cell body is single, large and spherical. It is centrally placed, pale, has one or two nucleoli. It does not contain centrosome. That is why the nerve cells do not replicate or multiply. You can see here in the cell body there are granular substance which are called nasal bodies. They are present in the cell body or the soma and the dendrites, but not in the axon or the axon hillock. You can see here the initial first part from where the process arises. This is called the axon hillock. Here no nasal bodies are found, and these structures, they are concerned with the synthesis of protein in the neurons, and their number is variable depending upon the condition of the neuron. The process by which the nasal bodies, they fragment and disappear due to fatigue or injury is called chromatolysis. Neurofibrils are thread-like structures found in the soma as well as the nerve processes. You can see here they are composed of microtubules and microfilaments, and they are specific to the neuron cells, not found in any other cell. If we talk of the dendrites, they are short and multiple, extensively branched. You can see here the shorter process, and these shorter processes, they contain both the nasal granules and the neurofibrils. They are basically the receiving or concerned with the reception of the stimuli, and they conduct the electrical signals toward the cell body. They receive the signal and they send it to the cell body, and then the cell body will further pass it to the or conduct it to the axon. While the longer process, generally longer process, which arises from the axon hillock, consists of a axis cylinder. It has exoplasm, mitochondria, and the axons, they end in the dilated part which is called the axon buttons or the axon terminals. And you can see here from the axon at the termination there are certain collaterals which arise, and the exoplasm is the cytoplasm which is present within the axon. and the function of this process is to conduct the impulse or the action potential away from the cell body. The size of the axon is variable. Some are a meter long and some are only a few millimeter. Their diameter is also variable. The larger the diameter, the faster is the impulse conduction, and the axon may be myelinated or it may not be myelinated. Only axons can have myelin sheath. The dendrites don't possess myelin sheath. You see here, the dendrites, the shorter processes, receive the signals and conduct to the cell body, where the signal is integrated, and this signal is then passed in the form of action potential to the axon terminal. The same thing here, you can see the axon hillock, and this is the initial segment of the axon. This is the myelin sheath. And now we will discuss about the axonal transport. The neurons, they are secretory cells, but they differ from other secretory cells because in these secretory cells the secretory zone is generally at the end of the axon, far removed from the cell body. जो बाकी secretory cells हैं उसमें वहीं पे synthesis होती है और वहीं पे secretion होती है, but here the secretory zone is far away and far removed from the cell body, and the apparatus for the protein synthesis is located for most part in the cell body, with the transport of proteins and polypeptides to the axonal ending by the axonal flow. So जो चीज़ें synthesize हो रही हैं cell body में, they are carried due to the axonal flow to the nerve terminal. And the cell body is very important because it maintains the functional and anatomical integrity of the axon, and if it is cut, if the damage occurs to the cell body, the distal parts will degenerate. Orthograde transport is the transport which occurs along the microtubules which are present in the axon, and they require two molecular motors, dynein and kinesin. These are the proteins responsible for carrying the substances from the cell body to the axon terminal. And in this there is a fast component in orthograde transport and a slow axonal transport. Two components. The fast transport occurs at about 400 millimeter per day, while slow axonal transport occurs at 0.5 to 1 millimeter per day. Retrograde, as the name is telling us, it is in the opposite direction from the nerve ending to the cell body. Occurs again along the microtubules at 200 millimeter per day, and the synaptic vesicles, they are recycled in the membrane, but some of the vesicles, they are carried back to the cell body. Opposite direction में जो transport होती है from the axon terminal to the cell body is called retrograde. Some materials, they are taken up by the ending by endocytosis, which include the nerve growth factor, and various viruses can also be transported by this mode. You can see here, यहाँ पे अगर आप देखें, the forward arrow is showing the orthograde transport, and the backward transport is called retrograde, and the transport requires microtubules which act as a pathway, and the molecular motors, you can see the enlarged view over here. The molecular motors, they carry the vesicle or other proteins or other cargo which has to be transported. They attach to it, and these motors, they carry the cargo all the way to the destination. The functional zones which are found in the neuron is, number one, as we talked previously, that the dendrites, they receive the electrical signals, the stimuli, they form the reception zone. You can see here, this is the reception zone, and this is the integration where all the signals they integrate, and ultimately the axon will form the conduction, and at the end is the transmission zone. The signal will be transmitted to the next adjacent neuron in series. Here, collection of the signal, function of the dendrites, forms the reception zone. The cell body integrates the incoming signals and generate the outgoing signal to the axon, and here in the axon it will conduct the impulse all the way to the nerve terminal. Now let's have a look at the different, how do we classify the neurons. They are classified in 3 different ways. We can classify neurons according to the number of poles they have, depending upon their function or the length of the axon. According to the number of poles, we divide them into multipolar, bipolar and unipolar. Multipolar has one axon, several dendrites, and they are abundantly found in the brain and spinal cord. The bineuron has one axon and branch dendrites and 2 poles, and they are least numerous kind of neurons, and they are found in particular locations like retina, inner ear and olfactory pathway. While the unipolar are also termed as pseudo-unipolar neurons, they have only one process which extends from the cell body. They are always sensory neurons, conducting the information towards the CNS. You can see here, this is a bipolar having 2 poles, one and 2 processes. One serves as a dendrite and the other serves as a axon. This is the pseudo-unipolar. You can see here it has 1 pole. One process is called the peripheral axon, and the dendrites, these are usually the sensory neurons. They receive the signal sent to the body, and and the body integrates the signal and sends a impulse back to the CNS. And the multipolar has a cell body. This is a prototype that we are going to discuss in our further lectures. The cell body with various poles called multipolar, smaller processes, the dendrites, the axon and the axon terminal. The other way the neurons are classified is according to the length of the axon. Golgi bottle type 1, in which the axons are long as those found in the CNS, and the Golgi bottle type 2 with short axons, and they are usually found within the cerebral cortex or within the spinal cord. Functionally, we classify them into sensory neurons. Sensory neurons are the one which carry the signals or transmit nerve impulses to the spinal cord. Then we have the interneurons in the spinal cord, gray matter, which integrates the signal and sends it to the motor neuron. This is the interneuron and this is the motor neuron. Motor neuron transmits the impulse away from the brain or spinal cord to the effectors like muscles or glands. Here, to sum it up, functional classes of neurons are motor, sensory and interneurons. Structurally, according to the number of poles, multipolar, bipolar and pseudo-unipolar. What are neuroglial cells? I told you that besides the neurons, which are the structural and functional units, there are other cells in the CNS, in the nervous system, which are called neuroglial or the glue cells or the support cells. They are actually non-neuronal cells of the nervous system. It is glia is a Greek word from glue, because these actually they serve as a glue. They keep the neurons bound in the bound form, and they are numerously found. They provide support for the nervous system functions, and this first term was described by Rudolf Virchow 1821 to 1902. They surround the neurons and support them. They themselves are not conducting cells. They do not conduct information, they only support the function of the neurons in various different ways. They are morphologically different from the neurons. They do not form dendrites or axons, and they are not functionally involved in the electrical signaling because they do not have the same membrane properties as neurons. The glial cells, they are found in both the central nervous system as well as the peripheral nervous system. They are of, so we can classify them as central neuroglial cells, those found in the central nervous system, and peripheral neuroglial cells, which are found in the peripheral nervous system. As regards the central neuroglial cells are concerned, they are astrocytes, microglia, oligodendrocytes, and ependymal cells. Now we'll take up each one of them separately. Astrocytes are star-shaped. Astro means star-shaped. They are the most abundant glial cells. They are present in all parts of the brain, and they have a unique intermediate filament protein which is called glial fibrillary acidic protein. They are of two types: astrocytes, fibrous and protoplasmic. The fibrous astrocytes are found mainly in the white matter, and they have an important role in the formation of the blood-brain barrier, while the protoplasmic astrocytes, they are found in the gray matter. They have end feet and they have dilated part. Their end feet are dilated that contact and surround the capillaries and arterioles throughout the brain. You can see here the protoplasmic and the fibrous astrocytes have long processes and end feet. These are the protoplasmic ones. Astrocytes, they form support network in the brain and spinal cord. They form blood-brain barrier. As you can see, their end processes are dilated and they invest surround the blood vessels. Their foot processes surround the blood vessels, so they maintain the chemical environment of the extracellular fluid around the CNS neurons. They regulate the cycling of the neurotransmitters during the synaptic transmission. You can see this is a general capillary and this is the capillary of the brain. The foot processes of the astrocytes, they surround the vessels so that things from the blood, the substances from the blood do not leak into the brain tissue, or substances from the brain tissue do not enter the blood. That is the form the blood-brain barrier. Microglia, as the name shows, micro means small. They are the smallest neuroglial cells. They are derived from monocytes and they are phagocytic in nature. They engulf and destroy the microorganisms and the cellular debris. They can migrate to the injured part or infected area of the CNS, and they can act as a miniature macrophage there. Oligodendroglial cells are the cells which provide myelination around the nerve fibers in the central nervous system. They provide support to the CNS neurons by forming a semi-stiff connective tissue between the neurons. You see here, this is the process of the oligodendrocytes, and it is investing the or surrounding the nerve fibers, providing them support. Ependymal cells are the cells which line the cavities of the brain and the spinal cord. They regulate the exchange between the CSF and the nervous tissue of the CNS. Where in the peripheral nervous system, we have the neuroglial cells called Schwann cells and satellite cells. Schwann cells are the cells which provide myelination in the peripheral nervous system around the nerve fibers. They have an important role in the nerve regeneration. They can remove the cellular debris during regeneration by their phagocytic activity. You see here, the myelin around the nerve fibers is formed by the Schwann cell. The satellite cells, they also provide support to the peripheral nervous system neurons. You can see here, surrounding the cell body of the neuron, and they help in the regulation of chemical environment of the extracellular fluid around the peripheral nervous system neurons. See, this is the astrocyte, the oligodendrocytes, the microglial cells, and the epidermal cells. This is all for today. Please go through this topic. We'll discuss it in the Zoom class. Allah Hafiz.